Hey fellow lab rats, this is Rebecca from the Lab Rat YouTube channel. In this video I'm going to be discussing lipids and lipoproteins as a part of my clinical chemistry lecture series. Alrighty, let's get started. Lipids are found in almost all tissues and have a vital purpose for the human body. Lipids are compounds that are soluble in organic solvents, not in aqueous or water-based solutions. So this means that they are going to be solid in water-based solutions. They serve a variety of essential functions. They're part of the structure within cell walls and in the transmission of nerves. They also help to produce steroids and assist with the immune system. Lipids also insulate and protect the body and serve as a store of energy reserves. The lipids that are clinically significant and those that we are going to be discussing today are fatty acids, triglycerides, phospholipids, and cholesterol. The first lipid we're going to be discussing in this lecture are fatty acids, which are the most basic form of lipids. Fatty acids are formed from a straight chain of hydrocarbons with a carboxylic acid group attached to the end of the chain. Fatty acids serve as the building blocks of two other lipid types, the triglyceride and the phospholipid. Within fatty acids, there are two different classifications, ones that are saturated and ones that are unsaturated. Saturated fatty acids do not have any double bonds within their hydrocarbon chain. Saturated fatty acids can be found in animal fats and are considered the unfavorable type of fatty acid as they can lead to atherosclerosis when eaten in high amounts in the diet. The unsaturated fatty acids have double bonds linking the hydrocarbon chain. They are found within plant oils and are generally considered better for overall health. So you can see both the saturated fatty acid and the unsaturated fatty acid chain in the picture on the right-hand side of the slide. So you see this is the unsaturated and there's that double bond, and this is the saturated, and you can see there are no double bonds here, so that's the difference. So for MLS and MLT students, you do not need to have these actual structures, chemical structures memorized. This picture is just here for your own information. Triglycerides are composed of three fatty acids attached to a glycerol molecule, as you can see in the picture on the right-hand side of this slide. So here's the glycerol, and then 1, 2, and 3 fatty acids. So the glycerol molecule is on the leftmost part of that picture, and then of course those three fatty acids are connected to it. So triglycerides make up a large part of the fat that is stored within the adipose tissues of the human body. Triglycerides provide structure for membranes and provide protection of the body's internal organs. A normal adult human should have a triglyceride value between 60 to 150 milligrams per deciliter within their blood. The next lipid we're going to be discussing are phospholipids. So these are very similar in structure to triglycerides. So the main difference is that instead of three fatty acids like in triglycerides, phospholipids have two fatty acids and a phosphate group. So if you see here, so here's the phosphate group, and then this is fatty acid 1 and fatty acid 2. The phosphate group is hydrophilic or polar, or attracted to water is what that means. And the two fatty acids are hydrophobic or nonpolar, which means that they're repelled by water. So this nonpolar and polar interaction allows the phospholipid to be part of the structure of the cell membranes and of surfactants in the lungs. So lung surfactant lowers the surface tension and thereby prevents the alveolar collapse during exhalation in the lungs. So you'll learn more about that later on in clinical chemistry. So this particular lung surfactant ability is particularly important in newborn babies. So again, we'll talk about lung surfactant testing later on. Cholesterol is another very important lipid, which is found only in human and animal cells. It has three main functions within the human body. So the first one is part of the membrane of cells. The second is a building block for the synthesis or the creation of steroid hormones. And thirdly, it makes up a component of vitamin D and bile formation. So there are two main sources of cholesterol, endogenous sources and exogenous sources. So endogenous cholesterol is made within the liver of the human or the animal. And this accounts for about 85% of all of the total cholesterol that's present within the body. So that's endogenous. Exogenous cholesterol is a cholesterol that comes from dietary sources. So this is cholesterol that you eat. It makes up the remaining 15% of the total cholesterol present within the human body. So overall, the majority of cholesterol in the body, so around 2/3 of it, is bound to ester. And 1/3 is just not bound to anything. It just floats freely around. Lipoproteins are formed from lipids and apoproteins. They are produced in the liver and the small intestine and function to transport lipids within the bloodstream. So there are 4 major types of lipoproteins, and these are chylomicrons, very low density lipoproteins, low density lipoproteins, and high density lipoproteins. And you see here, these have their abbreviations. So VLDL is very low density lipoprotein, LDL is low density lipoprotein, and HDL is, you guessed it, high density lipoprotein. The density of lipoproteins is compared to that of water, which is 1. So chylomicrons have a density of 0.95. Very low density lipoproteins have a density of 1.006. Low density lipoproteins have a density of 1.063, and high density lipoproteins have a density of 1.210. So you don't need to memorize these densities. This is only to give you an idea of where they got their names. So recall in the lectures on proteins and electrophoresis. And if you haven't watched that lecture already, please go ahead and watch it. It's also on my channel. So remember in that lecture that the migration areas on the protein electrophoresis are alpha, beta, gamma, etc. So lipoproteins can also be run through the electrophoresis system. HDL molecules go towards the alpha area of the migration pattern. VLDL molecules head to the area just before the beta region, and LDL molecules go towards the beta region. And now the chylomicrons stay near the well of the electrophoresis chamber, so at the origin. So I briefly mentioned the different classes of lipoproteins: chylomicrons, VLDL, LDL, and HDL. So let's talk about each of these a little further. So chylomicrons are a lipoprotein that is responsible for transporting exogenous triglycerides and cholesterol from the intestine to the bloodstream. So remember, exogenous means coming from what we eat. So these are triglycerides from what we have consumed in our diet. So once in the bloodstream, the triglycerides are removed from the chylomicrons to be used by the cells of the body, or if it doesn't need those, it is then stored in adipose or fatty tissue. Chylomicrons stay in the bloodstream for about up to 6 hours after a meal. When run via electrophoresis, chylomicrons stay near the origin of the well. Now, very low density lipoproteins, or VLDL, are lipoproteins that are responsible for carrying the endogenous triglycerides in the liver to the bloodstream. And again, endogenous is different from exogenous, so endogenous is just what is produced by the body. So the triglycerides are removed from VLDL and the remnant becomes a low density lipoprotein. Low-density lipoproteins, or LDL, carry cholesterol to the body's tissues to be used for the synthesis of needed substances such as steroid hormones or cell membranes. LDL cholesterol is considered the bad cholesterol, as large amounts in the bloodstream can lead to atherosclerosis. When run through electrophoresis, LDL migrates to the beta region. In an average adult human, LDL levels should be from around 50 to 130 milligrams per deciliter. So again, this is the bad cholesterol. So the lower this value is on the reference range, the better it is for the patient. High-density lipoproteins, or HDL, is a lipoprotein that is responsible for removing excess cholesterol in the bloodstream and then transports them to the liver to dispose of them. Because HDL removes excess free cholesterol, it is considered the good cholesterol. So in electrophoresis, HDL migrates to the alpha region. So in a normal adult human, HDL levels should be between 40 to 75 milligrams per deciliter. And again, since this is the good form of cholesterol, the higher that this value is on the reference range for patients, the better for it. So a patient has better levels of HDL if they have 73 milligrams per deciliter HDL level as opposed to 43 milligrams per deciliter. So you want a high value for HDL and a low value for LDL. Apolipoproteins are the protein portion of the lipoprotein complex. There are 5 main classes of apolipoproteins, and these are ApoA, ApoB, ApoC, ApoD, and ApoE. Apolipoproteins have several important functions within the body. So they help to transport lipids within the bloodstream, help to synthesize and break down lipoproteins, and can affect rates of various metabolic processes. Apolipoprotein A is the protein portion of high-density lipoproteins, or HDL. ApoA is produced in the intestine and the liver, first as a part of chylomicrons. Once in the bloodstream, ApoA transfers itself to the HDL molecule. Apolipoprotein B is the protein commonly associated with low-density lipoproteins, or LDL. There are 2 types of ApoB. So ApoB 100, which is created in the liver, and ApoB 48, which is created within the wall of the intestinal tract. Physicians can compare the ratio of ApoA to ApoB to determine a risk of heart disease. So a ratio of greater than 1 is considered desirable, and anything less than 1 is associated with heart disease. Apolipoprotein C is the protein commonly associated with very low density lipoproteins, or VLDL. And this apolipoprotein is created mainly within the liver. So apolipoprotein D has a minor role with high density lipoproteins, or HDL, although not much is currently known about this apolipoprotein. Apolipoprotein E is formed in the liver as well and has a role in a lot of things, so LDL, VLDL, and HDL. So like with everything in clinical chemistry, of course we need to know normal reference ranges. So when I say reference ranges, again, what I mean by that is that these are the expected ranges in a normal human population. And these vary just very slightly between each individual laboratory. So we have total cholesterol, HDL, LDL, and triglycerides. So these can be run as individual tests in the clinical laboratory, or they can be run as something called a lipid panel. So a lipid panel includes all of these. So total cholesterol, HDL cholesterol, LDL cholesterol, and triglycerides. So that's considered a lipid panel. So the doctor can just order that panel on this patient, or they can order each 1 or 2 of these tests or 3 of these tests, whichever. They can be ordered individually if needed. So total cholesterol's normal reference range is 140 to 200 milligrams per deciliter. HDL is 40 to 75 milligrams per deciliter. And so remember, HDL is the good cholesterol, so you want a higher range HDL. LDL's normal reference range is 50 to 130 milligrams per deciliter. And again, recall that this is the bad cholesterol, so you want a lower range LDL level. And then lastly, triglycerides should be 60 to 150 milligrams per deciliter. Coronary artery disease, coronary heart disease, and atherosclerosis are diseases that involve abnormal lipoprotein levels. So this happens when coronary arteries struggle to supply the heart with enough blood, oxygen, and nutrients. So cholesterol deposits or plaques are almost always to blame for this. So if you look at the picture here on this slide, it shows at the top here a normal artery. Everything is clear in that artery, allowing normal blood flow. Now, the artery at the bottom of this picture on the right-hand side of the slide has a buildup of lipids in it, or plaque, which narrows the blood flow in it. So it narrows the artery, and it causes a blocking the flow of blood within that artery. So this increases the risk of myocardial infarction, which we know already is the scientific or the medical name for a heart attack. And so pause just a second here and think for me for a second. So do you remember what the best chemistry marker is for heart attacks, right? So if you have not watched the enzymes and cardiac markers lecture already, go ahead and do that. But if you have, you should know that the answer to that question is troponin, right? So troponin is the most sensitive and specific cardiac marker for a heart attack or AMI. So risk factors for coronary artery disease include eating a fatty diet and also obesity, smoking, having high blood pressure, and also if the patient has a sedentary lifestyle. So also hyperlipidemia, which is an excess lipid in the blood, can be a risk factor for it, and additionally a family history of coronary artery issues. Hypolipoproteinemia occurs when there are too many lipoproteins circulating in the bloodstream. So patients can either have primary or secondary hyperlipoproteinemia. Primary hyperlipoproteinemia happens when there is an overproduction of lipid synthesis due to an inherited defect or an acquired problem. Secondary hyperlipoproteinemia occurs as a result of an underlying disease, such as diabetes mellitus, kidney or liver disease, or pancreatitis, which is an inflammation of the pancreas. So there are 6 main types of hyperlipoproteinemia. So there's 6 main types of hyperlipoproteinemia, so that's an increase of those lipoproteins, and there are 2 types of hypolipoproteinemia. So hypo meaning it's a decreased level. So this is a decreased level of lipids in the blood, and both of these you do need to know. So these disorders are Abetalipoproteinemia and Tangier disease. So Abetalipoproteinemia is an inherited disease that prevents the body from absorbing fat and also fat-soluble vitamins. So in this disease or this disorder, there's a decrease of LDL cholesterol and VLDL due to the absence of ApoB, so apoprotein B. So patients also have a decreased level of cholesterol and triglycerides in their bloodstream. So peripheral blood smears of patients with Abetalipoproteinemia often have what we call acanthocytes. So this photo on the left-hand side of the slide shows a peripheral blood smear with acanthocytes on it. And you'll learn about these in hematology. So let me see if I can circle them here. So this is an acanthocyte, and this is an acanthocyte, and this one is as well. And you see these are all normal red blood cells. So acanthocytes are in patients that have Abetalipoproteinemia. So it's important to note that you can have acanthocytes and not have this disorder. It's just one thing that's associated with this Abetalipoproteinemia. So Tangier's disease is a rare genetic disorder that due to the absence of ApoA, there is a decrease of HDL cholesterol in the bloodstream. So again, HDL cholesterol is considered the good cholesterol because it removes excess cholesterol from the bloodstream. So because patients with Tangier's disease have low levels of this HDL, this means that they have a higher risk of developing coronary artery disease. In addition to a lipid panel, which again includes total cholesterol levels, HDL, LDL, and triglycerides, there is something called a standing plasma test that can be performed. A standing plasma test is performed by observing a fasting plasma for an abnormal appearance after a refrigeration period overnight. So after that refrigeration period, chylomicrons appear as a creamy white layer on the top of the plasma. VLDL gives a milky or lipemic appearance throughout the sample. LDL gives an orange tint to the sample, and HDL doesn't have any effect on the sample, so no turbidity or lipemia. So that's the standing plasma test. The Abel-Kendall method is the reference method for total cholesterol. So again, when I say reference method, this is the clinical chemistry assay that all other tests are compared to for this particular analyte, which is total cholesterol. So Abel-Kendall is the reference method for it. So this is a chemical method that is very complex and time-consuming. Now the Lieberman-Burchard method, this is also a chemical method for total cholesterol, and this uses hazardous chemicals such as chloroform to produce a result. So because of the potential danger and time required of chemical methods, total cholesterol assays are now most commonly performed using an enzymatic method, which we'll talk about on the next slide. So in the enzymatic total cholesterol assay, the enzyme cholesterol esterase is added to the patient's serum or plasma, and this cholesterol esterase hydrolyzes the cholesterol esters. So remember that this is the bound cholesterol, so this is stuff that's bound in ester form. So this hydrolyzation unbinds it, so everything in that sample is then going to be that free cholesterol now. So then another enzyme called cholesterol oxidase is added to that sample, and this cholesterol oxidase oxidizes the free cholesterol to produce both cholesterol and peroxide. So lastly, the next step of this enzymatic method is the enzyme peroxidase is added to it, and this is used to measure the peroxide. And the amount of peroxide is proportional to the amount of total cholesterol present within the sample. So back in the day, this was all performed manually, but this is now a test that is performed on a chemistry analyzer. So you put the sample on the chemistry analyzer aliquots out the serum or plasma and does this entire reaction within the confines of the analyzer, and then it pops up with a result. Direct HDL assays are commonly performed in the clinical lab. So when I say direct, what I am referring to is that it's actually measuring the particular analyte. So in this case, it's actually measuring the HDL. So specific detergents are used to prevent enzymatic reactions in every other lipoprotein than HDL within this assay. So once these detergents are added, reagents are then added to quantify the HDL that is left over. Now, in most clinical laboratories, a patient's LDL level is a calculated result. So when the LDL is calculated, we call it an indirect LDL level, and an indirect LDL level is included in a lipid panel. So the following formula is how LDL is calculated. So you take the patient's triglyceride level and divide it by 5, then add the patient's HDL level. So this result then is subtracted from the patient's total cholesterol level. So this gives you the indirect LDL level on the patient. And again, the chemistry analyzer is not directly measuring the LDL level, it's from a calculation, and this is why we call it an indirect LDL level. Now it's important to note that this calculation is only valid if the patient has a triglyceride level under 400 milligrams per deciliter. So are there patients that have triglyceride levels over 400 milligrams per deciliter? And the answer to that is absolutely yes. So what happens when this is the case? So what happens when the patient has a higher than 400 milligrams per deciliter triglyceride level? So in that case, the indirect LDL level cannot be calculated and a direct LDL must be performed. So an immunochemical method must be performed to measure the patient's direct LDL level. So I have created another video of me working through a couple of indirect LDL calculation problems, and I will go ahead and link it in the description of this video, so please check that out. Triglycerides were once measured via a chemical method that used either alcohol or chloroform to extract the triglycerides out of a patient's sample. And because of the potentially dangerous nature of this test, clinical laboratories now perform an enzymatic method to measure triglycerides in the blood sample. And again, these are all performed on chemistry analyzers. So hardly anything manual is done in clinical chemistry these days. The sample gets put on the analyzer and it gives you a result. Lipid samples have specific requirements. So generally when ordered, a lipid panel or lipid profile includes a total cholesterol level, HDL cholesterol level, triglyceride level, and a calculated LDL cholesterol. So as a review, this is an indirect LDL level because it is being populated by a calculation. So the patient needs to be fasting for 12 hours before a lipid panel or lipid profile is drawn. If the patient is getting just a triglyceride level performed, a 12-hour fast must be performed as well. Now, if the patient is only having a total cholesterol level test performed, fasting is preferred but is not required. So these tests are generally performed on a green top or a gold top tube, depending on what laboratory that you may be working in. Alrighty, so that concludes our lipids and lipoprotein lecture. If you like this video, go ahead and give it a like, and please remember to subscribe to my channel for more educational laboratory content. And as always, if you have a question, please feel free to write it in the comments section. I will check it and I will answer it as soon as I can. Alrighty, until next time.